So welcome back. We have been discussing on carbohydrates. We already saw the structure, functions, properties of different carbohydrates. We saw that carbohydrates are divided into classified into monosaccharides, oligosaccharides, and polysaccharides. We also saw the properties of monosaccharides and disaccharides, the functions they perform, etc. Okay. So here what we are going to discuss is the tests conducted for carbohydrates. As for tests, we can have two kinds of tests. One is qualitative test, the second is quantitative test. Okay, qualitative tests are done for detecting the presence or absence of a molecule in a sample. Okay, so when we are doing qualitative tests for carbohydrates, what we intend to do? We intend to detect the presence or absence of carbohydrates, or in general sugars. Okay, so if you are getting a positive result for the qualitative test, we can say that the sugar is present. Otherwise, it is negative. Okay, but quantitative tests on the other hand, quantitative tests are done for estimating the amount of the particular sugar in the sample solution. Qualitative tests cannot actually give us the proper quantity of sugar in a sample, but this one, quantitative test, usually helps to detect the amount, or sorry, what you call estimate the amount of sugar in a sample solution. Okay. Now, when we discuss about carbohydrates, when we have a sample solution, we don't know what is there in that. Okay, we just have a sample solution. We need to identify what is a biomolecule, or what is the what you call yeah, the macromolecule present. Okay. Now, first what we need to do, we need to identify whether it is a carbohydrate or not. Okay, so to test whether it is carbohydrate or a non-carbohydrate, we can conduct malicious test. Okay, malicious test, when we conduct malicious reagent is there, we just need to add it, and if we get a positive result for that, we can say that the carbohydrate, if the sample solution contains carbohydrate. If otherwise, carbohydrate is absent, so it is a non-carbohydrate. But since it is not present, it is not supposed to be it is not mentioned in your syllabus. I'm not going into detail the principles and all. Okay, once we are sure that the sample solution contains carbohydrate, what is the next step? We need to know whether it is a reducing sugar. Okay, reducing sugar, again, before that, one test which we are already familiar with, the starch test, iodine test. Okay, we just need to add iodine solution to sample, and if we get a positive blue black coloration. Okay, so what does it indicate? It contains starch. Okay, otherwise it is a non-starch, right? So next one we can have is the Benedict's test. Benedict's test help us to identify whether it is a reducing sugar or a non-reducing sugar. Okay, all the monosaccharides, they are reducing sugars because they all have a anomeric carbon, or what you call carbonyl carbon free. Okay, a free carbonyl carbon or anomeric carbon. If it is free, obviously free for bonding. Okay, free for formation of what you call this one, oxidation. So if such a all such molecules, sugars, will give a positive result for Benedict's test. Okay, if it is not, it is non-reducing sugar. Okay, now under the reducing sugar, obviously all monosaccharides are reducing sugars. Apart from the monosaccharides, we can see that some of the disaccharides like lactose and maltose are also reducing sugars, because these two molecules, they do have, these two sugars, they also have, what is it, an free anomeric carbon. So it also gives a positive result for Benedict's test. What about sucrose? Sucrose is a non-reducing sugar, so it doesn't give a positive result for Benedict's test. So we can easily identify a reducing or differentiate between a reducing sugar from a non-reducing sugar with the help of Benedict's test. Okay, almost it is the Fehling's test also can help in differentiating between monosaccharides and disaccharides. Okay. Now, coming back to the next one, it is Barfoed's test. Barfoed's test helps us to distinguish between reducing monosaccharide and reducing disaccharide. Okay, so when we are getting reducing sugars, okay, we get a positive result for a particular sample. Okay, so we can be sure it is reducing sugar, but we cannot be sure whether it is a monosaccharide or disaccharide. So to be sure whether it is a monosaccharide or disaccharide, we can go for Barfoed's test. Okay, now under Barfoed's test, we can very well distinguish monosaccharide and disaccharide. If it is a monosaccharide, we have to identify which is it, aldoses or ketosis. Okay, how do we distinguish? We distinguish it with the help of the Seliwanoff's test. I hope it is clear. So this is a sequence. Okay, malicious test, obviously it is not mentioned in your syllabus. Then Benedict's test, Fehling's test, Barfoed's test, and Seliwanoff's test. Okay, now we can see the Benedict's test. Benedict's test, what we do, we do get Benedict's reagent as such. Okay, Benedict's reagent we take 5 ml and almost equal amount of the sample solution is added. We mix it well and boil it. Okay, boil it for 2 minutes and then let it cool for some time. Okay, now what happens? The entire solution, if it is positive, okay, if it is positive, that is if it contains reducing sugar, obviously what happens? It gives a precipitate. Okay, the color of the precipitate depends upon the concentration of the sugar solution. With the least concentration, it is green precipitate, and with maximum high concentration, it is red precipitate. Okay, so in the absence of a reducing sugar, blue color of the reagent as such it remains. Okay, so we can see how the color is. This is it. Okay, so we have taken the sample solution, we have added what you call a Benedict's reagent, and if we get such a coloration, you can see there is no precipitate here. Okay, this means it doesn't contain any reducing sugar. Okay, but when such a coloration develops, you can see precipitate is there. Okay, if a precipitate is formed, either green or yellow, orange or brick, we can be sure that it is reducing sugar. Okay, green or yellow shows very little amount of sugar is there. How much we cannot actually identify with the help of this one. We may have to go for calorimeter and all. Okay, so we can be sure that there is a sugar. Okay, this is moderate amount, and if it is brick red color, obviously there is a large amount of reducing sugar. Okay, so that is an observation we can get. Now, what can we infer? The color of the precipitate gives an idea about the concentration, but we cannot be sure that this is this much. Okay, exact amount cannot be identified. You can see that reducing monosaccharides, glucose, fructose, galactose, and mannose gives a positive reaction. Okay, you can see this is the sugars. Okay, different kinds of sugars. You can see lactose, fructose, glucose. All these are, you can see the precipitate is given. Sucrose, you can see it is blue color, isn't it? Plain color, just like this one. Okay, so there is no sugar, but no reducing sugar. Okay, now coming to the principal side. Benedict's reagent, it contains copper sulfate, sodium citrate, and sodium carbonate. Okay, um, actually this particular test, it takes the advantage of the reducing ability of the monosaccharide or reducing sugars. Okay, so what happens? The sugar, it acts as an, yeah, it gets oxidized, and during the particular process it reduces cupric to cuprous. Cupric ion or cupric ion containing compounds, they are blue in color, while cuprous compounds, they are red in color. Okay, so that is how we can see the change. Okay, so what happens is it contains copper sulfate, where copper is in cupric form. Okay, it contains sodium citrate and it contains sodium carbonate. Right, now what happens is, in the presence of sodium carbonate, after sodium carbonate, it provides an alkaline medium. In the alkaline medium, what happens? Glucose gets converted into enediol. Okay, enediol. Right, now at the same time, copper sulfate in solution in aqueous medium, it gets dissociated to form cupric ions and then combines to form cupric hydroxide. Okay, cupric hydroxide do have the tendency to get precipitated, but due to the presence of sodium citrate in the Benedict's reagent, the cupric hydroxide, it remains unprecipitated. Clear? So there is cupric hydroxide and there is enediol. Now enediol is a very strong reducing agent. So what happens? This enediol, it reduces cupric. Okay, cupric hydroxide, it is blue in nature, color. Okay, when cupric hydroxide, it gets converted or it gets reduced to cuprous oxide, it becomes red in color. Right, so this is how we can actually determine the presence of the glucose or any other reducing sugars. Is it clear? Okay, now next is Fehling's test. Fehling's test, almost it is the same. It is used to determine the reducing sugars and aldehyde. We can perform it in a different set of sugar solutions. Okay, here unlike Benedict's reagent, you can see there are two solutions: solution A, Fehling's solution A, and Fehling's solution B. Fehling solution A contains copper sulfate pentahydrate, where copper is again in Cu2 plus form. Okay, and solution B, it contains potassium sodium tartarate, that is Rochelle salt, and sodium hydroxide in water. So what we do is we take the sample solution, we add these two sample, this solution A and solution B in equal amount, and then we boil it. That is what we do. Okay, and this helps in testing the detection of monosaccharides. Right. Now, what happens here? Okay, this is what happens. Okay, when there is no reducing sugar, it gives a blue coloration. When we have the what you call carbohydrate, present, okay, that is monosaccharide reducing sugar present, obviously it gives such a brick red color. Okay, after boiling, that is it. Okay, it gives a positive. Now this is the reaction which goes on. So the D-glucose, it converts cupric to cuprous, reduces cupric to cuprous. At the same time, it gets oxidized at this position, C1. Okay, it becomes gluconic acid. Right. Now Barfoed's test. Barfoed's test, as already mentioned, it helps in differentiating between the what we call reducing monosaccharide from reducing disaccharide. What we do is we take a Barfoed's reagent, 5 ml, and add the sample solution, especially the um, almost a 0.5 ml, and then mix well. And then what we do is we boil it. Okay, and we can see that the red precipitate clinging onto the bottom of the test tube. You can see here. You can see this xylose, glucose, fructose, lactose, sucrose, etc. Right, but here you can see these ones give the red precipitate like this. Okay, clinging onto the bottom. Right, you can see even though lactose is a reducing sugar, but since it is a disaccharide, it doesn't give a positive result for Barfoed's test. So we can differentiate uh, reducing disaccharide from reducing monosaccharide. So okay, so here a red precipitate clinging to the bottom of the test tube. You can see it indicates the presence of monosaccharide, and it is the positive result is given by monosaccharides only, like glucose, fructose, galactose, mannose, etc. Now, reducing property owes to the carbonyl group. Barfoed's a reagent contains copper acetate in acetic acid. So, unlike the Benedicts or Fehlings, here it is, you can see it is acidic medium. The other one was alkaline medium. Okay, you can see here it contains sodium hydroxide in alkaline. Here it is sodium citrate, sodium carbonate. Okay, that is again alkaline medium. So, here it is Barfoed's, it is acidic medium. Okay. Now, how we can differentiate? What are the differences between Barfoed's and Benedict's test? Simple thing. Barfoed's test takes place in alkaline medium, in acidic medium, while Benedict's test takes place in alkaline medium. Okay. In acidic medium, monosaccharide enolize much more readily than disaccharides. Okay. That is in the Barfoed's test. And these endiols, it reduce cupric ions released by copper acetate. So here what happens is Barfoed's reagent gives a positive result in monosaccharide. But when we keep the particular solution for quite a longer period of time, maybe even the reducing disaccharides may give a positive result because it takes some time. That is the difference. Okay, in it monosaccharides gets converted into indiol. Disaccharides also gives may uh, like get converted to indiols, but the time taken is different. Okay, so the first one to give an answer will be monosaccharide. Okay. Now, the second, so we have already identified this is a monosaccharide. Now we have to identify whether it is an aldose or ketose. So this is done with the help of Seliwanoff's test. Okay, Seliwanoff's test gives positive result in ketosis. In aldoses gives a negative result. Okay, so what we do, take 5 ml of Seliwanoff's reagent and we add fructose solution. If it is fructose solution, okay, we have a keto solution here. So we have fructose solution and we boil it. Okay, a cherry red precipitate is what is the positive result. So you can see such a kind of cherry red coloration develops, and this is due to the presence of ketosis. Okay, uh, and this is actually a dehydration reaction due to the hydroxyl groups of the sugar. You remember the sugar contains it is a polyhydroxy compound, isn't it? So because of the hydroxyl groups, it gives a positive result, right? And Seliwanoff's reagent, it contains resorcinol in dilute hydrochloric acid. What happens to ketosis in hydrochloric acid or in acidic medium? It gets dehydrated. Okay, and the aldoses gets converted into hydroxymethyl. Uh, what ketosis it gets converted to hydroxymethyl perfura, and this condenses resorcinol to form a red color complex. Okay, so it is actually because of the like dehydration of ketosis by hydrochloric acid that actually results in the formation of perfural compound that condenses the resorcinol to form a red colored complex. Okay, so this is it. Right. So we have seen the different kinds of qualitative tests to detect the presence of uh, carbohydrates.